Good afternoon, or evening, or morning, or whenever you're watching this. My name is Claire Stevenson, and today I will be summarizing the biochemical pathways that we have learned thus far in our nutritional biochemistry class of Dr. Calloway's Fall 2024 version. So the pathways that we have learned so far are glycolysis, which is important for bringing glucose into the cell and allowing it to be rapidly metabolized into energy, either into pyruvate or lactate, depending on the presence of oxygen. Gluconeogenesis, which is important for maintaining blood glucose and creating glucose from non-carbohydrate sources. The citric acid cycle, which is really important for generating reducing equivalents or the electron transport chain, which will work in combination with oxidative phosphorylation to make massive amounts of ATP production in the presence of oxygen. Then we'll talk about glycogenesis and glycogenolysis, which are really important for regulating blood glucose levels by either converting glucose into glycogen or converting glycogen back into glucose. Finally, we'll wrap up by talking about the pentose phosphate pathway, which generates NADPH, which is important for anabolic reactions such as fatty acid synthesis, as well as creating ribose 5-phosphate for nucleotide synthesis. So let's get into it. First, we're going to talk about glycolysis, which has some really important irreversible steps. So the first step, third step, and final step are all irreversible. So the first step is bringing glucose into the cell, either by and then phosphorylating it with hexokinase or glucokinase, and hexokinase has a greater affinity for glucose. But since we're phosphorylating glucose, you need a phosphate group, so we'll convert ATP to ADP, and then that will give us glucose 6-phosphate. Glucose 6-phosphate is incredibly important in several of the pathways later that we'll be talking about. But just for now, I'll mention that glucose 6-phosphate is important for glycogenesis and glucogenolysis, and the pentose phosphate pathway. Now, when the enzyme is in black here, then that means that is a reversible step, meaning that this enzyme is used both for glycolysis and gluconeogenesis. So when glucose 6-phosphate is converted to fructose 6-phosphate by phosphoglucoisomerase, it doesn't matter which way the reaction is going, it'll always be done by this enzyme. When fructose 6-phosphate is created, it's important to mention that this is also an important intermediate later in the pentose phosphate pathway, But this is the second irreversible step in glycolysis performed by the enzyme phosphofructose kinase 1. And that step also requires a phosphate group, so an ATP to ADP conversion to create fructose 1,6-diphosphate or FDP. From here, it is converted by fructose biphosphate aldolase to create GATP and DHAP. So this is where we start to rearrange the actual carbon molecule. So this whole time we've had 6 carbons, and here's where we switch, we create 2 3-carbon molecules. And then DHAP is converted into GADP, which then is converted by glyceraldehyde phosphate dehydrogenase into 1,3-biphosphoglycerate. This step is going to generate, an NADH from an NAD. And since this is going to be happening twice, once from the GADP molecule, and then second from the DHAP molecule that's converted into GADP, then we'll generate 2 NADHs from this step. Next, glyceraldehyde phosphate dehydrogenase will create 1,3-biphosphate glycerate. Then phosphoglycerate kinase will create 3-phosphoglycerate, which will be used later in the pentose phosphate pathway. And then this step is going to require is going to generate 1 ATP per molecule, so it'll be a net amount of 2 ATP. And then 3-phosphoglycerate is converted by phosphoglycerate mutase into 2-phosphoglycerate. So we're simply just moving which carbon the phosphate group is on, judging by the 3-phospho and the 2-phospho. And then enolase will create phosphoenolpyruvate. And the final step of glycolysis is also irreversible and will generate an ATP molecule using the enzyme pyruvate kinase to create pyruvate. So from to summarize glycolysis, we will end up with 2 pyruvate molecules and a net amount of 2 NADH and 2 ATP molecules. Unless oxygen is not present, then we'll make lactate into ATP. Because if we're using 2 ATP here and generating 4 ATP here, then it'll be a net amount of 2 ATP. So from here, if we need to make glucose from a non-carbohydrate source, one potential pathway is actually basically the reverse of this, where we will generate pyruvate, we'll convert pyruvate all the way back up into glucose. And so most of these steps in gluconeogenesis are the same, but there are a couple that are different, and it will be these 3 steps that are irreversible in glycolysis. It's important to note that gluconeogenesis is only occurring in the liver and kidneys, and it is where the majority of glucose production occurs in ruminants. So the first step to get around this irreversible step of glycolysis in gluconeogenesis is going to be taking pyruvate into the mitochondria. And this will be done using the microbial protein carrier, excuse me. And then pyruvate carboxylase will convert pyruvate into oxalacetate. And the reason why pyruvate is taken into the mitochondria is because that's where the pyruvate carboxylase enzyme is to convert pyruvate. And then while in the mitochondria, oxalacetate will be converted into malate, since that's the only thing that can actually exit B mitochondria. Because then when it's back in the cytoplasm, then oxalacetate will be created from malate again. And then this allows oxalacetate to be converted by phosphoenolpyruvate or PEP carboxylase back into phosphoenolpyruvate. And from here, enolase will convert it into 2-phosphoglycerate, which will take 1 GTP molecule per pyruvate and 1 ATP molecule per pyruvate. So that's 2 and 2. And then the step is the same. The step is the same, other than the fact that instead of generating energy, we'll be using an energy molecule in the form of ATP for every pyruvate molecule. Same thing for this step. It's going to be performed by the same enzyme, except for instead of generating energy, we'll be using energy. And that'll be, again, for each pyruvate molecule, so times 2. Then once we get back up into these last 2 irreversible steps, the reaction from fructose 1,6-diphosphate to fructose 6-phosphate will be performed by fructose 1,6-biphosphate and release a phosphate group, which makes sense because we're using a phosphate group when we're in glycolysis, and we'll be releasing a phosphate group when we're going back up into glucose in the gluconeogenesis cycle. And then we will convert fructose 6-phosphate into into glucose 6-phosphate using the same enzyme as glycolysis, phosphoglucoisomerase. And then glucose 6-phosphate will be converted by glucose 6-phosphatase into glucose. And then again, since we used an ATP over here, then we'll be releasing a phosphate group in gluconeogenesis. So most of the gluconeogenesis is taking place in the cytoplasm, except for a little bit in the mitochondria, and then all of glycolysis is occurring in the cytoplasm. So from here, we're going to go deeper into the mitochondria, which is shown by this orange line. And it's important to note that oxalacetate into malate is a reversible reaction that also occurs in the citric acid cycle right here. So when we are starting a citric acid cycle, we'll be using pyruvate, which was generated in glycolysis, and pyruvate will be converted into acetyl-CoA by pyruvate dehydrogenase. So it'll go from a 3-carbon molecule into a 2-carbon molecule, and acetyl-CoA will combine with oxaloacetate, which is a 4-carbon molecule, uh, and citrate synthase will then combine these two molecules to make citrate, which is a 6-carbon molecule. So 2 plus 4 equals 6. And then this is the first irreversible reaction in the citric acid cycle. So as you can see, most of these arrows are reversible except for citric citrate synthase, and then these two steps right here, which we'll talk about shortly. So from here, citrate is converted into oxine, oxinate. This is really testing my English. By the enzyme oxintase, and then oxintase will also convert it into isocitrate. And this whole time, it's going to remain a 6-carbon molecule. But once isocitrate dehydrogenase converts isocitrate into alpha ketoglutarate, then we'll lose a carbon dioxide molecule and we'll convert from a 6-carbon molecule to a 5-carbon molecule. And this right here is indicative of aerobic respiration, and also why when bread is being baked, then it will rise because all the air bubbles that make it light and fluffy, like a good bread, well, I mean, you can have like a very dense bread as well, but like let's just take sourdough, for example. There's a lot of like air bubbles in sourdough. Even when you cut it, you can still see where those remnants of air holes were, like in Swiss cheese, and that's because of this carbon dioxide production. And so it's this metabolic process that allows bread to rise. Anyways, enough of the side tangent. This step also creates NADH from an NAD plus molecule. And then from alpha ketoglutarate, we will convert using the alpha ketoglutarate dehydrogenase enzyme into succinyl-CoA. And just like this irreversible step, we'll release a carbon dioxide molecule and we'll convert from a 5-carbon molecule to a 4-carbon molecule. And then we'll remain a 4-carbon molecule all the way back until this step when we create citrate again. So this step also creates another NADH, and then succinyl-CoA can then go on to be converted by succinyl-CoA synthase into succinate, which will generate a GTP. And then succinate is converted into fumarate using succinate dehydrogenase and also creates another energy reducing equivalent, FADH2. And then fumarate is converted into malate with the enzyme fumarase. Malate is converted into oxaloacetate using the malate dehydrogenase enzyme and creates another reducing equivalent, NADH. So then all these reducing equivalents, which are shown in green, and these green arrows are pointing in one direction down to the next phase, which is oxidative phosphorylation. So these reducing equivalents will first be used in the electron transport chain, which are these enzymes that are in the inner mitochondrial membrane. And 3 of the 4 complexes have pores that go all the way through this lipid bilayer to pump hydrogen atoms into the intermembrane space to create a high hydrogen proton gradient. Complex 2 does not have a pore, but it is used for electron transport. And then these 2 mobile units, coenzyme Q and cytochrome C, both act as electron carriers in this electron transport chain to help create this gradient. So this biological process is what makes aerobic respiration so much more efficient than anaerobic respiration, because simply by using the chemiosmotic theory, which is basically that if there's a high concentration of hydrogen atoms in this intermembrane space, then they're going to want to naturally, through osmolarity pressure, go back into the mitochondrial matrix. And then the ATP synthase is going to use that concentration gradient to its advantage to then make mass amounts of ATP. So complex 1 will accept electrons from NADH, and NADH will be converted into NAD+. A hydrogen atom will be pumped through, and then the electrons will be passed on to coenzyme Q from complex 1. Complex 2 will accept electrons from FADH2 to create FAD. And Like I already said, it doesn't have a pore that goes through that inner mitochondrial membrane, so it won't pump any hydrogen atoms into the intermembrane space, but it will pass on its electrons to coenzyme Q. Coenzyme Q will then take the electrons from complex 1 and complex 2 and pass it on to complex 3. And then that energy will allow complex 3 to pump a hydrogen atom through. The electrons will then be passed to cytochrome C, which will pass them on to complex 4. Um, and this will allow more hydrogen atoms to be pumped through. And the final electron acceptor is oxygen. So all of this electron passing is possible because of oxygen. Like I already prefaced, this is aerobic respiration, which means that there's a presence of oxygen. And so oxygen is incredibly electronegative, which means it's very, very greedy for electrons, especially two more electrons, which is exactly what it gets from this reaction. And so all these complexes and enzymes, whenever they get the electrons, then they're going to be excited with energy, and that's what allows them to pump hydrogens through. But it's also this like excitement from the electrons is like a hot potato. They don't want it. It's too much energy potential for them. But oxygen really wants it. It's very greedy. So oxygen is what's driving this entire electron transport chain to carry the electron through to then make H2O, water, very safe molecule. Um, but that electron transport chain that's powered by the electron force creates this huge hydrogen gradient, and the hydrogens will then be pumped through the ATP synthase through these C units. It'll attach to one of the C units, and then each time a hydrogen atom is pumped through, it will rotate uh, this synthase 120 degrees, and it will create one ATP molecule. Now, technically, it takes three hydrogen atoms to create one ATP molecule because ADP and a phosphate group will bind to one of the beta subunits. And then once it's gone a full rotation, then it'll be released as ATP. So if one hydrogen atom rotates it 120 degrees, then it would take three hydrogen atoms to rotate it 360 degrees. So it technically takes three hydrogen atoms per ATP molecule, but one hydrogen atom is what generates an ATP molecule. So then under perfect conditions, this will generate a maximum of 38 ATP. Because think back to all the reducing equivalents that we've made so far in glycolysis and in the citric acid cycle. We've made a lot of NADH and FADH2, and each NADH molecule will pump three hydrogen atoms through, which will then go on to create 3 ATP, and each FADH2 molecule will pump two hydrogen atoms through, which will then go on to create 2 ATP. So once you do all this math, then you get a max amount of 38 ATP under perfect aerobic conditions, excuse me. This 2 ATP from glycolysis uh, is considering these extra 2 ATP that's generated when oxygen is present and we create pyruvate, not lactate. So this is the powerhouse. This is why the mitochondria is called the powerhouse of the cell, is because of this process right here. The oxidative phosphorylation process allows these once upon a time ancient aerobic microbes, mitochondria, to actually metabolize um, products using oxygen and using that electronegativity of oxygen to make really effective uh, energy synthesis. So from here, we're going to be converting back into the cytoplasm. When we talk about glycogenesis and glycogenolysis. So the first step in glycogenesis, which is an anabolic process. So we're going to be creating something. We're going to be creating glycogen from glucose. Is the first step is technically going to be also the first step of glycolysis when we're bringing glucose into the cell and then phosphorylating it with glucokinase or hexokinase. And glucokinase is only present in the liver, whereas hexokinase is only present in our muscles. So it depends on which cell type you're talking about as to which type of enzyme will be used. Um, so glycogenesis is going to occur after we after an organism eats an animal, human, mammal, ruminant, whatever you want to call them, because After a meal, insulin levels are going to increase. And then the insulin is what's going to be driving this glycogenesis process. And we'll talk about it more later when we talk about glycogen synthase. Uh, but the reason why we want to start storing glucose as glycogen right after we eat is because we don't need all that energy from glucose right away. So in order to actually, have better energy partitioning, we're going to store glucose as glycogen until it's needed later. So, like I said, this first step is going to be the phosphorylation of glucose. And just like in glycolysis, one ATP molecule will be used in order to phosphorylate the glucose molecule and trap it in the cell. And then we'll create glucose 6-phosphate, which, like I said in glycolysis, is a huge central point to a lot of these metabolic pathways. And we'll talk about that later again. But then from here, phosphoglucomutase is going to convert glucose 6-phosphate into glucose 1-phosphate. And then UDP glucose pyrophosphorylase will convert glucose 1-phosphate into UDP glucose and release 2 phosphate groups. And then we get into the irreversible step of glycogenesis, which is the conversion of UDP glucose into glycogen using the glycogen synthase molecule, and this will release a UDP molecule. So when insulin is present, then it will allow for the dephosphorylation of glycogen synthase by the protein phosphatase 1 enzyme to be in the active state. So when glycogen synthase is dephosphorylated, it's active. And then from here, we have alpha 1,4 glucosyl. And then using the glycogen branching enzyme, we can make glycogen because you can have an alpha 1,4 linkage or an alpha 1,6 linkage. So in order to get from glycogen back to glucose, we're going to go through the glycogenolysis process, which is a catabolic pathway, meaning that we're destroying catabolic catastrophe, glycogen into glucose. And so then this is going to be stimulated by the hormone glucagon during times of fasting when glycogen needs to be broken down in order to have glucose available. So, after a meal, you have high glucose because you've eaten, but 5 hours later, when you're hungry and you're not quite able to eat your next meal, then you're going to have high glucagon, which means that your body's going to be breaking down glycogen into glucose. So in order to convert into glucose 1-phosphate, glycogen phosphorylase the enzyme is going to be used, and a phosphate group will also be used. So glucagon is going to allow for the phosphorylation of glycogen phosphorylase by protein kinase A, and that's when it's active. So when glycogen phosphorylase is phosphorylated, it's active, whereas when glycogen synthase is dephosphorylated, it is active. And then this is the only irreversible step in glycogenolysis. And then from glucose 1-phosphate, we'll use the same enzyme as we did in glycogenesis, phosphoglucomutase to then create glucose 6-phosphate. And if we're in the muscles, then this is going to be the end because then glucose 6-phosphate will then be shuttled into the glycolysis pathway and be converted into energy from there. So if we remember, going back up, following this green line, all the way up here into the first step of glycolysis, glucose 6-phosphate, excuse me, can then be used either in gluconeogenesis or glycolysis, but either way, shuttled back to this main pathway to create energy. So follow the green line back down. However, if we're in the liver, then glucose 6-phosphatase will convert glucose 6-phosphate into glucose, which can then be shuttled into the bloodstream and goes to whatever tissue it is needed in. Another fate for glucose 6-phosphate is to enter the pentose phosphate pathway. So following that green arrow down here. So this first phase of the pentose phosphate pathway is the non-reversible phase. So all of these arrows are only pointing in one direction because these reactions are non-reversible. And the oxidative phase is important for creating NADPH, which is used in fatty acid synthesis, cholesterol synthesis, and uh, lutein reduction. So glucose 6-phosphate will be converted into 6-phosphogluconolactone by glucose 6-phosphate dehydrogenase. And this will create an NADPH. From here, 6-phosphoglucolactone will be converted into 6-phosphogluconate by glucolactone hydrolase. And then 6-phosphogluconate dehydrogenase will create ribulose 5-phosphate. And this is where carbon dioxide is released, and we convert from a 6-carbon molecule, which is what we've been, into a 5-carbon molecule. Um, sorry, I was just checking something. But this step will also generate another NADPH. So we'll create two NADPHs in the oxidative phase of the pentose phosphate pathway. And then ribulose 5-phosphate is incredibly important for nucleotide synthesis. And so this pathway can actually stop here if that's what's necessary. Ribulose 5-phosphate can enter the non-oxidative phase if we are needing more glycolytic intermediates, which are down here, or if we want to make ribulose 5-phosphate from glycolytic intermediates such as fructose 6-phosphate and glyceraldehyde 3-phosphate. So just as review, if we go back up to glycolysis and gluconeogenesis, the enzymes that will, excuse me, the products that we'll be creating at the end of the pentose phosphate pathway is fructose 6-phosphate and 3-phosphoglycerate. So these are both important intermediates in glycolysis and gluconeogenesis. And if we need to, we can use these intermediates to create our R5P for nucleotide synthesis. So back down to the non-oxidative phase of the pentose phosphate pathway. The enzyme ribulose 5-phosphate isomerase will create ribose 5-phosphate, and ribulose 5-phosphate epimerase will create xylose 5-phosphate. And both of these are going to remain 5-carbon molecules. And then from here, these two molecules can be converted using transketolase, which will require magnesium, into cetoheptylase 7-phosphate and glyceraldehyde 3-phosphate, which, like I've mentioned, is that intermediate in glycolysis and gluconeogenesis. And then transaldolase will convert these two molecules into etherose 4-phosphate and fructose 6-phosphate, which, as you remember, fructose 6-phosphate is an important intermediate in glycolysis and gluconeogenesis. And then, so this part gets kind of confusing because, we've got to have arrows pointing in both directions because it's a reversible reaction. But that's why I just kind of overlaid this green outline to kind of make it simpler. But xylose 5-phosphate and erythrose 4-phosphate will be converted using transketolase into fructose 6-phosphate and glyceraldehyde 3-phosphate. And then these two can be shuttled into the glycolysis and gluconeogenesis pathways. So I think hopefully that covers everything that it is necessary for our exam question in order to understand how all these pathways are connected, what end products they make, how their intermediates are related, how the reducing equivalents are related, because all of these pathways are constantly going on inside of all of our cells. And the scary and really cool thing about biochemistry is like there's not one end or beginning. So I did this in a very chronological order in a very organized fashion, but within our bodies, all of these things are happening at once. And so it's really important to understand not only how they're happening, but also how they're interconnected and how they impact metabolism, glucose levels in our blood, energy availability, and energy expenditure. But yeah, I hope you enjoyed and have a good rest of your day.